One of my favorite quotes is, "Life is not measured with the number of breaths we take, but by the moments that take our breath away." This is certainly one of them for me, and I hope it becomes one for you. Let me start by sharing a picture that I myself find breathtaking. This is a picture of our Earth taken from space. And life on this Earth, the only one that we have, is supported by a fundamental process that I am sure many of you are familiar with: photosynthesis. Photosynthesis starts with the absorption of light. And what I'm trying to do here in this video is taking you through a leaf, then deep in the cell membrane, very, very, very deep, just at the nanometer scale, where we will find the molecular machinery that supports the absorption, conversion of energy. This photosynthesis process is done not only by plants, but also by algae and by photosynthetic bacteria. And we have been learning a lot about this process for many, many years. But our understanding at this molecular level is only improving in the last few decades, because we have been able to go down to this nanometer scale, understand how these molecules behave, how the vibrational motions at this level cooperate to transfer the energy that they are absorbing from the sun and convert it into the chemical energy that supports all life on Earth. And by this I mean that I think it's around 118 billion tons of biomass. If you can imagine, this is about as if nature is producing a chart building every second. So it is not surprising that scientists have tried to understand what is happening at this molecular level in photosynthetic systems and in many other biological systems. Now, life on Earth, the Earth is all about 4.6 billion years, or 4.6 thousand million years, just to make sure that we're talking with the same billions. But life on Earth only started a billion years after that, and it started with photosynthetic bacteria. Nowadays, we use these systems to understand. And when I say we, I am a theoretical physicist, but I work closely with a group of scientists that are trying to understand these molecular processes in photosynthetic systems. What you see in this picture is first the image of how you see photosynthetic bacteria in the lab. This is a macroscopic image of each individual bacterial cell. And this is an atomic force microscopic image of the membrane of that cell. And it's in that membrane that we find these biomolecules that I am so interested in understanding how they behave. This is an illustration of those biomolecules that you find here. We call these light harvesting antennae. And the name is because they are the ones that capture the photons that are coming from the sunlight, transfer energy within each of these molecules, then transfer energy to another molecule, then transfer energy further until it reaches a particular center where chemical energy conversion starts. Now, the same process happens, but not just in bacteria, but in algae. Life of algae started even later, 2.1 billion years ago. And the process is very similar, but it has different structures of how different structures to absorb light. Here again, you see a macroscopic image of each of these cryptophyte algae cells. This is an image of the membrane of the cell. And then here, we don't have in between images yet, but we do have an illustration of the molecules that you will find in these structures that will absorb that light. Now, what I'm going to do is I would like to pass around just some 3D models of the biomolecules, which I'm going to explain and will be my toys to explain the theory that we do. I will pass this around, and I will ask you please not to break them. It was very hard to print this 3D. This was done about 5 years ago when it wasn't so popular to print 3D models of this. This has been amplified. This is the length scale of these molecules is around 5 nanometers. This has been amplified more than 1 million times for you to see. Please don't break them. And these two are particular instances that I have focused on understanding because they have different structures, they have different chemical components, yet they behave very similar. And this shows us a bit about the beauty of nature, evolving very different structures, yet utilizing the same principles for transferring energy. You can see the videos as well here moving. Now, just so that you have a picture of what is this photosynthetic process at this first molecular level, these first steps, here is an illustration, and I will break down the understanding of this process as I go along in this talk. So what is going to happen is that light is going to be absorbed by one of those light harvesting antennas that you have. This light harvesting antenna, you can imagine as many chlorophylls bound together by a protein. Now, when you have many chlorophylls together, they no longer work individually. They cooperate to absorb the light. What it means is that rather than each chlorophyll absorbing individually, they now join efforts, so they interact via electronic interactions, to then absorb light that otherwise they wouldn't absorb individually. This energy is transferred between different antennae until it reaches this reaction center where charge separation, so the chemistry of photosynthesis starts. Now, different organisms and different biosystems utilize this principle, photosynthetic systems, and one common feature is that when the light is very dim, so a very, very low intensity lights, almost every photon that is absorbed generates a charge separation in this reaction center. This is much higher than any man-made solar cell. So this is an efficiency of around 90%, which means that out of 100 photons, 90 will generate a charge separation event at the reaction center. Now, photosynthesis needs for this process to be so efficient because overall photosynthesis, as we go to larger scales, is not going to be so efficient. So it cannot afford to lose the energy that it absorbs right at the beginning. Now, it's not surprising then that we have been interested in understanding what are the physical principles that support this very efficient process at this level. We have a pretty good understanding. We know that energy is not so easily dissipated in these systems. The transfer of the energy happens very, very quickly within a molecule and towards another molecule. And when I say very, very quickly, we're talking about femtoseconds or picoseconds. Now, if you want to have an image in your mind, many of you will know what this scale means, but if you just want to have an image of this, a femtosecond will be 1 million times faster than the blink of an eye. So it's a process that happens in a scale that it seemed unreachable until a few decades ago. But nowadays, and this is one of the most important developments in this field, we have access to very sophisticated both experimental tools and theoretical tools that are allowing us to understand those processes at those length scales, a few nanometers, 1 billion times smaller than the thickness of the hair, and at time scales so fast that before we could have not thought of this to be possible, but we now can. Some of these experimental processes are this optical multidimensional spectroscopy, a very fancy tool, many different lasers shining a sample of photosynthetic bacteria or cryptophyte algae, where I have millions of those molecules, but they are so diluted that they behave almost as if they were independent. But we have also now the opportunity to investigate each of these molecules individually. Different groups, different experimental groups are trying to isolate these molecules so that we can understand how the physics of each of these molecules vary within the systems, but also so that we can have access to these very fast processes just at that individual level. I am interested in that level. And the reason I'm interested is because while we all agree that these processes happen very fast, there is still some ambiguity on the quantum nature of the processes that happen at this scale. Now, we know for sure that as we go down to the atomic level, the physics rules that dominate the world that we see around us, we need to redefine it. How we describe what happened at that level is given by the rules of quantum mechanics, the mathematical tools, the conceptual tools that we have there. I'll tell you a little bit about that, and then I'll tell you what is the aim of the research that I do. I'd like to highlight in particular that I'm interested at this particular level because I have been able to join efforts with an experimental group at Glasgow University, who is the group of Luca Sapienza, and together we are trying to isolate these molecules, and we're trying to find unambiguous evidence that indeed these fast processes function in a way that the quantum mechanical behavior cannot be neglected at any point. Now, my focus on what I will present here will be mostly about photosynthesis, and then I will show you how the same principles, the same quantum principles that we're starting to understand in these systems, do also operate in other molecular systems. So the understanding that we're gaining from nature at this molecular level in photosynthesis is an understanding that is giving us tools to understand well beyond that. It's kind of telling us what we maybe could have guessed. Nature doesn't reinvent the wheel. Once nature has found a solution for what is efficient, for what is the best way of transferring, for what is robust, it takes those principles and distributes it across different scales and across different systems. It has had more than 3.6 billion years to evolve and find the solutions. Now, this is not, I am a theoretical physicist. The early career researchers that I work with are theoretical physicists, but this is not an endeavor only of the physics community. In fact, it is more an endeavor that more traditionally was associated to the physical chemistry and the biophysical community. But because the way we understand the problem is so different, the way we have different tools from these different communities, this is now a full interdisciplinary endeavor to really understand the physical behavior, quantum physical behavior of these molecules. What I believe is happening is that we are facing, at this time, what people would understand as a paradigm shift in the understanding of our molecules that support life on Earth. And this paradigm shift is going to change the way we can manipulate life processes at that molecular level. So we are starting to find a very different way to understand the quantum science that we can find in those biomolecular systems, and therefore that opened the door for using many new tools from quantum science to affect those biosystems. And eventually, when these communities interact more often, and the communication and the understanding from the different perspectives becomes more cohesive, then we will be able to say a little bit more about the biology of that quantum regime. Now, as a theoretical physics, my aim is to build theoretical and computational approaches that will allow me to help accelerate that paradigm shift. That is my purpose. And to do that, the core of what I do is centered on an idea that we call theory of open quantum systems. What this means is, who has the molecule? One molecule. There. So if you see that molecule, the molecule will have the yellow background, which represents the proteins, and it will have some structures that represents the chlorophylls. So if I move in this direction for a moment, whoever has the green molecule, this is a 3D version of the one that I'm presenting here in 2D. And in this molecule, these curvy structures represent a protein, and these black structures represent the pigments, which absorb the light individually when they are far apart, but collectively if they are interacting via their location here in this protein. These chlorophylls, or these different pigments, in this case, these are not chlorophylls, these are phycoerythrin, so it's another kind of pigment that will absorb light. From a chemical perspective, they are made of many different molecules, many different atoms, and they have a multitude of electronic states that you can investigate. From the perspective of open quantum systems, which is the perspective that I work, I see those systems, those electronic systems, as different electronic states, different electronic possible motions, quantized motions, as I'll explain in a moment. And everything is around the nuclei motions that comes from the internal part of the pigments, the nuclei motions that come from the protein. I see that as an environment. And the reason we do this is because in the lab, we normally don't have access to all the possible motions that describe this system. So we have optics, which means that we are addressing the electronic motions in these molecules. So these open quantum systems allowed me to reduce a little bit the complexity of understanding the physics of this system, while at the same time, being able to build an explanation of what we are observing in the lab. Again, I try to, and when I say I, it's I and the research group that I lead. We try to integrate different methodologies from all these different disciplines to build a cohesive theoretical framework to understand this quantum behavior, to predict that what you might observe in particular experiments, and to be able to advance this understanding. Now, let's go on to the meat of the problem. The main question that we're trying to address in this field is whether these systems support quantum processes. What do I mean by that? Let's go back to our molecule. So what is happening here is that this molecule is absorbing light, which I represent here with this wave. We know that this light is quantized, so the light is coming in packages that we call photons, and this system is absorbing these packages of photons. How is it absorbing this? Is already a quantum mechanical process that we all agree. When this light is absorbed, what is being affected by the absorption of this photon is the electronic motions of these chromophores or pigments. I will use different names as they are known in the literature. Now, what is the change of these electronic motions as these photons are absorbed? That is what is explained by quantum mechanics. Just to give you an insight, a picture of the quantum mechanical processes, I'm going to make an analogy with classics, although we know quantum and classics do not necessarily have a direct correspondence. But the closest concept to understand what is happening with those chromophores is the interference that you see when you put two stones in a very calm lake. If you put two stones in a calm lake, you form waves that propagate. And if these waves interfere, you will see some regions where you have black, some regions where you don't have. And what this represents is you have different motions of the matter here. Now, at the molecular level, before I go into that, the way we describe this is a superposition of the possible motions that come from the wave on the right and the possible motions that come from the wave of the left. At the quantum level, something similar can be used to describe what is happening there. But we don't have waves really there as what we have in the water. The waves that we have there is more about the probabilities of how we will affect the system and the motions that it will have. Now, I'm going to enter a little bit of the math, but don't worry, it's not a lot, but it will give you the main insight that then will allow us to understand what is happening in nature. If you imagine that now that molecule that is moving around is reduced to only two of those pigments bound to the protein, the different colors of these pigments means different light that they can individually absorb. And this on the left represents that if it hasn't absorbed, it's just in the ground state, and if it has absorbed this pigment, then it will be excited, and I'm going to call this 1. And on the right, it represents the green pigment. If it hasn't absorbed light, it's on the ground state, and if it has absorbed, it's on the excited state, and I'm going to call this the one of the left. it happens that when these pigments are interacting, the light that they absorb is neither red nor green. They absorb lights at different frequencies. And for me to understand what is the light that they will absorb, I will use that concept of interference of the different states. From a more physical perspective, what is happening here is that there is a probability that when a photon is absorbed by this molecule, that this will be excited, but then because of interactions with the other pigment, it will get de-excited, but this one will get excited. Essentially, they are sharing the excitation, but the way they are sharing this excitation is similar to that interference pattern that we have. It's not just sharing by passing one another. I like tennis. I play tennis a lot. And if you want to imagine a little bit, if you play tennis, this sharing is as playing doubles. The ball is coming, and you are working in a unity, if you play tennis properly. You are working in a unity to respond to that ball that you can imagine is the photon. This is a way of imagining this process. The main idea that I want to give here is that this collective behavior, we already know that we can only explain it with the rules of quantum mechanics. So there is no doubt that to absorb sunlight, these molecules are fundamentally using quantum mechanics 101 or 100, however you call it, at university. Now, the way we know this is that nature has evolved a variety of light harvesting antenna that actually covers the whole visible spectrum. We have organisms absorbing near the infrared, which is the purple bacteria, the light harvesting 2 complex, which is the yellow molecule that you're passing around and that you will return to me at the end. And we have in the other extreme the cryptophyte algae, which is the green molecule that you are passing around as well. And while they absorb in very different frequencies, they have very different structures, the physical principles that govern that behavior are the same. So one of the main problems that we face here is that in order to understand this system from the theoretical perspective, we really need to be very accurate. It's not enough just to have an insight into how they should absorb. So part of the work that we've been doing, and many other groups around the world, is how to describe as accurate as possible these absorption properties of these molecules, in general, the whole spectroscopy. In my group, currently I have, sorry, I forgot here the name, but this is Charlie Nation, and this is Hallman Oscar Getson. These are 2 early career researchers working with me. And one of their main aims is that they wanted to be able to predict the absorption properties of these molecules with a very high level of accuracy. And what you can see here, T2 and T3, means 2 different approaches, 2 different levels within the same approach, and T3 is the level of more accuracy. So what this is illustrating is as we increase these levels, our accuracy in predicting this is the absorption for this light harvesting complex, the one in purple bacteria, the one that you have in your hands, is becoming better. So we're very happy about this because we now have a theoretical framework and a computational framework that allows us to predict with a pretty good accuracy the absorption properties of systems as large as that molecule. Now you will see, but it's just one molecule. This is not a large system. In reality, this is a system that has 27 chromophores. And if you were to compute the full dynamics in a computer, it will take many, 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 many days to compute it accurately if you take into account every single atom and every single nuclei. So with the approach that we take, we're able to predict this with an accuracy that I will show you in a second why we need such an accuracy. Now, what this represents as well is in that ring that you have, in that LH2 light harvesting antenna, it has 2 rings, it has 2 sets of chromophores or pigments. You have one band that is the red band, and then one band that is the blue one. The red one here has the chromophores strongly packed. The blue one, the chromophores are far apart. What this is telling us is that individually, each bands will absorb differently. And this is what we predict in the theory and what it is observed in the experiment. The blue band absorbs around 800 nanometers, but as they become interacting even closer and closer, like in the red band, they absorb at around 850 nanometers. In nature, what happens with this system is that light will be absorbed first by the blue band, so the 800 nanometers, and then it will transfer energy from here to here. So physically, it will be from the blue band to the red band. And if you want to see it in this figure, energy will go from here to here. Now, this figure is also telling us another story, and is that as these pigments interact more strongly, then the frequency that they will absorb will move in that direction. Now, this is very important for one of the problems that we are trying to address from the theory viewpoint, and it's the following. In order to understand the molecular and the physics and the quantum physics behavior of these individual molecules, what we do in the lab, in the molecular system in the cell, they are on the membrane. But doing the experiments on the membrane are not as simple as we would like. So what experimental scientists do is that they work with these molecules. They have solutions of these molecules in which they have used detergents and many other chemicals to make sure that these molecules are isolated from the membrane, and then in a solution they are separated, so you have individual membranes. And this has given great insight into the physics of this molecule. But one of the things that I kept wondering for many years, I've been studying this problem for about 15 years, and just one parenthesis, I remember having a conversation with a colleague many years ago. He had spent about 20 years understanding one particular problem, and I was at the time a grad student, and I told this colleague of mine, I don't understand how you can be 20 years studying the same problem. At the time, I was a PhD student, and I was just thirsty for knowledge, so I just wanted to learn so many things. But now, I fully understand this colleague, because I fell in love with the physics and with the understanding of these molecules. I close the parentheses. So one of the main problems that have been in my mind, I thought, great, we can isolate these molecules, and we can have solutions of hundreds and thousands of these molecules. But these are not how the molecules are in the biological system. Are we changing anything when we isolate them, when we take them outside from the cell? And if we do, if we're changing anything, what exactly is that we're changing? Now, this is not an easy question to answer, because it means that we really need to have a very high precision when we measure them in and out the cell membrane. And we need to find conditions that are actually comparable. So if I go and measure in the membrane, it's not the same as measured in the solution, because in the membrane, not only they're constrained, they're also interacting weakly with other molecules. So the problem wasn't easy, but it was always in my mind, what have we changed when we moved them out of the membrane? And most importantly, going back to the quantum properties, are we changing anything associated to the quantum properties? So this question was in my mind when I had my first PhD student, but we couldn't address that question. That was around 2008. But recently, there were some experiments from the group of a colleague in MIT, Gabriela Slough-Cohen. Gabriela and her team had the same bugging question in their mind. So what is the impact, essentially, of the membrane on these quantum properties of this system? So they designed a system that looks like the biological system. So this is kind of an artificial membrane. It has the same, let me say, material that you will find in a cellular membrane. So this is a lipid bilayer. And Gabriela and her group are able to embed these light harvesting complexes in this artificial membrane, but they are so far apart that they can investigate them as if they were individual molecules embedded in the membrane. So I was very excited when I saw this and I saw the results, because then I said, okay, now we can understand what is the possible effects. Gabriela was predicting that when you take this molecule out of the membrane and when we investigate them on the detergent, on the solution, yes, we get some insight into the quantum properties of these molecules. We get some benchmark of what is happening there, But when we take them out, we make some modifications that actually change in about 30% the energy transfer within the molecule. And 30% doesn't seem just a little thing. 30% is significant change. So I was really, okay, I really have to explain what is happening here. So last year, Chantelle Kulkarni joined my group, and I proposed her, okay, look, this is a problem that I have had in my mind for a very long time. Can we solve it? Can we try to understand this? And she said, oh, yes, let's work on this. And I was very excited because the things that I presented to you are results that we obtained 2 days ago. So you are the first ones to read about it, apart from my collaborators. So this is very exciting because this is the following. So what you see here on the left are the experimental results from Gabriela. The green and the gray curves represent, the green is when they measure the absorption of these molecules on the artificial membrane, and the gray one when they measure it in a solution of isolated proteins in detergent, in water, in a solution that is when they do the experiments. Now, some of you would say, well, they're very similar, like nearly the same, so you only have a little bit of a difference here and a little bit of a difference here. Well, it happens that it is consistent that you will see a shift in the absorption towards the right, so towards longer wavelengths, going from the membrane to the disk. From one perspective, you would think that this change, which is just a few nanometers, about 4 nanometers, 5 nanometers in other systems, is very small. You might think, oh, this might be within the error of the experiment, no? So then maybe that's not a real difference. Well, these results are reproducible, which means other labs can go and measure the same, and then we'll come up with the same conclusion. There is a shift in the absorption of these molecules. Now, this shift is very significant because it means that the interactions that are happening within the molecule, so if you have the yellow molecule, there is interactions in that red part, are changing, and are changing significantly to affect the absorption. So we join efforts with Chantelle, with Harman, who has developed that very accurate setup to be able to predict this absorption properly. And if you think about it, we need to make sure that we are not predicting something that is changing so that the next approximation will then give me something along these lines. So Harman has made that possible, Harman and Charlie and other members in my group. And this is our theoretical comparison. This is one level of approximation, but we now have the more exact level of approximation. And if you see here, we are predicting a similar shift. In order to do this work, so here the dotted line is the absorption in detergent, and here our black line is the absorption in this membrane disk. This line just represents the light that the blue band in your molecule, the one that you're seeing at the moment, will emit. I'll talk about this later. The most important is that we are able to predict this. You see Chantelle is here smiling? Well, that was the smile when we talked a couple of weeks ago when we first saw this. But I'll show you something more. She was even more smiley last week. Why? Because we were able to predict that that small change, what looks small, actually represents that significant change in the energy transfer within the molecule as it was measured for the group of Gabriela in MIT. So she was able to demonstrate by doing these accurate computations that energy transfer from the blue band to the red band was being increased by 30% when it is on the membrane. So it was decreased when we put it on the detergent. And 30%, as I said, might seem nothing. But in terms of the quantum efficiency of transfer within the molecule, it's a significant step. So we were very happy because when we compared the numbers that she was predicting, her numbers, Chantelle was in the detergent 625 femtoseconds, and on the membrane, 140 femtoseconds. Gabriela's numbers are experimentally measured 607 femtoseconds, 875. We were super excited. Now you can say, oh, but you are quite far, 40 femtoseconds here, you are still 35 femtoseconds. Here you are 45, femtoseconds. You are still far. Yes, we're not exactly the numbers. We cannot match the experiment exact, but we can match the qualitative difference that we're seeing there. So this has been really good news for us, because now I can then focus on understanding what are the changes in the quantum properties. So while it's been great that from the experimental viewpoint, it is possible to measure all these properties of the molecules isolated, and that has given us, and will continue giving us, important insight into the understanding of energy transfer and quantum processes there. One thing that I'm starting to understand with this small calculation is the fact that in the biological environment, the quantum properties are actually even more robust. Very, very counterintuitive for what you would expect. Because on the membrane, on the cell, things there are moving all the time. This is a system completely out of thermal equilibrium, but with many different random motions where you would expect that some of these quantum properties might actually be more fragile there. At least that was the thinking. But what we're seeing here is that in the membrane, which produces some pressure, lateral pressure on the on the light harvesting complexes that you see, you actually see that that maintains and enhances that quantum chemical structure of the system. So now, Chantal will continue smiling because we will continue exploring what are the possible quantum effects now when these molecules are analyzed actually in the membrane environment that we have. And what I expect is that probably other quantum effects will be enhanced in that environment. Now, I'd like to move on to something a little bit more controversial. So what I have told you so far, if you were to talk to other scientists from other disciplines, from physical chemistry, other colleagues in California, Greg Engel in the United States, Greg Scholes, Graham Fleming, well, so many people all over the world investigated, we will all agree. We will say yes. So we understand that as far as absorption processes are, quantum mechanics and emission processes, quantum mechanics determines. So we have no problem there. But now we enter into a very difficult terrain here. And it's the terrain in which we are going to investigate whether there are other quantum processes, more dynamical, that actually happen on these molecules. That is where some disagreement still exists, when the issue is not settled. And I'm hoping, or is my aim, that the work we're doing at the moment and the interactions we have with this experimental group in Glasgow will help us advance our understanding in that direction. Now, one thing again that we will agree is that whatever is happening in these molecules is helping us to rethink the way electronic motions and vibrational motions cooperate to transfer energy. So at this very molecular level, you can imagine this as a sea of an electronic cloud in the system, and it's not just the one from one pigment, the other one is coming collectively from all the pigments, but you also have these nuclei motions individually in each chromophore and coming from the protein. And one thing that we're understanding is that there are two kinds of different motions there. Some motions that will maintain that collective behavior, so things moving in a synchrony, but some other motions that will tend to localize and destroy that synchrony. And it's an interplay between these two things that promotes energy transfer in the system, but also it will play a very important role in what we understand for non-trivial quantum effects in these systems. To give you like a picture in your mind so that you get away and you have an analogy, it's not exactly like that, but that you might have an analogy for this, is the following. So I talked about those interference patterns that is a way of seeing what is happening when these things absorb light collectively. Now, as soon as light is absorbed, energy will be transferred within the molecule. So electronic excitations of these chromophores will start displacing around the molecule until it is transferred into another molecule. But because of all those vibrational motions that are thermal, near thermal motions, these interference patterns will be destroyed, just as it happens when we are very happy throwing our little stones, and then someone comes with the motor in the sea and destroy that pattern. Similarly, this is a way of illustrating what will happen at this quantum mechanical level. But I'm going to show you how is that we illustrate it. This is a simulation that was created by a former PhD student in my group, Dr. Richard Stone's now doctored. And it's a simulation that you can have access if you visit my website in the outreach part. And this simulation is showing me the following. So in the 3 panels that we have here, panel A, B, and C, what I'm going to show you is different situations in which I have a different level of vibrational, let me call that noise for now. It's not really noise, but let's call it for now. And by this, I'm illustrating the vibrational motions, whether they are intramolecular for each of the chromophores, whether they are the protein, whether they are collective of all the molecule, all those possible vibrational motions, I am illustrating there as this motion for that protein. In this first panel, what I'm doing, let me just put this for a second, is that energy, this is hypothetical. I'm starting energy in this red chromophore. Somehow I managed to make sure that the absorption only happened here. And then after that photon is absorbed, energy, the electronic excitation energy starts being transferred around in this molecule. This energy transfer is going to be dominated mostly by the interactions, the electronic interactions between these chromophores, but you have some level of motions, of these vibrational motions, but I'm going to say that that level is very weak. So what you see, or what you expect, or how you can understand this, is that energy actually is being shared at the same time, just as in those interference patterns, but these different molecules. Oops, before we go into there. Don't go so quickly. What happened here is that energy was being shared by the whole molecule. And the sharing of that excitation lasted for quite a long time before energy was localized in the final chromophore, which is my target chromophore. When I ran that many, many times, so for this video it was run many times, it predicts that the average transfer time from here to here is about 4.2 picoseconds. Now, I'm going to do the same, but I'm going to assume that that level of noise is even higher. So those vibrational motions are more intense and therefore are affecting more the energy transfer within the molecule. However, it's even higher in the left panel. So here is like an intermediate level. It's a level in which is comparable to the strength of the interactions that are promoting energy transfer between the chromophores. So what we see is the following. You see some kind of sharing of energy, which is represented by light being illuminated in different chromophores at the same time. But you see that at every time, you can actually find excitation here, not so much here. In this intermediate level, then we see that the average transfer time is around 1.4 picoseconds. In this final level, I really have these things moving very high, very strongly coupled to this electronic system. And if you see here, energy is mostly localized at every time in each of these chromophores. And then it starts again, but no, we don't want that. What I want to illustrate here is that with this high level of noise, vibrational noise, we see that the energy transfer, the transfer time is around 3.8 picoseconds. What is the lesson that this experiment is giving us? Can someone tell me? I'll tell you. It's telling me that in the middle, I seem to find the best regime for energy transfer. It's a regime in which I have some level of vibrational noise, but not so much that it will destroy the cooperative behavior of those electronic states in the system. So I am in that middle regime. From the physics viewpoint, what this is happening is that this system has has different motions and different degrees of freedom and different interactions, but all the interactions are comparable in strength. So no one interaction is very weak in comparison to the other ones. And because of that, you have competing effects. And the best regime is precisely when those energy scales are comparable. And it seems that while there is a wide range of those interactions, these molecules do not remain static. In time, they have structural changes. Those structural changes happen in a much longer time scale than the picosecond time scale that are comparable here. But still, they will affect the energy transfer in the system. But even within those structural changes that they naturally exhibit, you still see that these molecules, they definitely don't operate here. Here is too quantum for it to be efficient. They operate between these two regimes. And this is one understanding that I would say many communities would agree. Now, the question is, how long and how quantum are those collective motions when you have so much noise destroying them? And that is one of the problems that we've been trying to address. Now, from the experiment, the information that we got, it was that the role of those vibrational motions in the energy transfer was more active than we thought before. We always knew from a long time ago, that you need the vibrational motion to transfer energy. Without it, you don't transfer the energy. But what we didn't know is how exactly those vibrations were being affected as energy was transferred. Now, there is a simple explanation of why we didn't know, because we focused on what was happening to the electronic degrees of freedom. When we measured in the lab, it was the optics. We measured the vibrational responses of this, but mostly to understand the vibrational structure. But we didn't focus on what was happening to the vibrational motions. Now, I've got a small group. So my group has always been about 5, 6 people rotating throughout the years. And I compete because science is competitive. And by this I mean I compete to find interesting explanations with groups that are much larger. Why is this important? Because I really need to focus on what questions people haven't addressed that I can give a good insight that complements what other people are doing. So in other words, I don't really want to compete with the other scientific communities. What I want to do is complement the insight that they are giving. And this thinking is what led us to then focus on the question, but from a different perspective. So the experiment was already giving us some insight that vibrational motions were very important in the energy transfer here. So at the time, I had a PhD student and a postdoc, and I said, look, why don't we focus on understanding the dynamics? So most of the time we focus on understanding the dynamics of the electrons in this. Why don't we focus on understanding the quantum dynamics of some of the vibrational motions that are involved in the transfer? And then let's see where that takes us. It has taken us to a very important and interesting field, which is the vibronic field. One thing that we understand now is that really that interplay between electronic motions and vibrational motions in this molecule is rich, is super interesting. It's giving us a completely different insight into how these motions cooperate to transfer energy. It's helping us to see these molecules not just as something that transfer energy, small thermal machines, small vibronic machines. And therefore, that leads us to many different experiments and many ways of thinking and implementing this. Now, again, let's go back to that example in which we have 2 of these chromophores only, and they're interacting. But now the system, the way I want to see it, is in this way. So we have these 2 chromophores that has those energies, as we described before, but each of these chromophores is interacting now with an oscillator. Now you will be, why am I representing an oscillator in this way? It's because that oscillator is also quantized as the electronic system. So now my main mechanism for photosynthesis is not just my electron degrees of freedom, it's also some important quantized motions of the nuclei. And I'm going to call this collective vibronic mechanism. Why do I mean by that? I mean by that those oscillations that might come from molecule on the left and molecule on the on the right, those oscillations might not be directly coupled because they are local. But through the interaction with the electronic degrees of freedom, then some correlations are found between this on the right and this on the left. And this collective vibronic mechanism is, I would say, one of the most interesting aspects that we are starting to understand more about photosynthesis, how it is used, how we prove it, and how it leads to quantum effects that are not trivial. As I said, so one of the most important things that I did was to focus on these vibrational degrees of freedom. We focused as well on what was happening to the electronic degrees of freedom, but we wanted to understand what was happening to those vibrational degrees of freedom in interaction with the electrons. And that has been my work for the last 10 years, and it's the work of many communities around the world as well now to understand those vibrational motions, those vibronic mechanisms. And it has led us to propose how this vibronic mechanism is very important to direct energy, to have non-trivial quantum behavior, to synchronize on motions within the molecule, and to actually control the current that you might extract from these molecules. Now, in the remaining of my time, which is 10 minutes, I just want to give you an overview of why it is so rich and where this is leading us. So as I said, this is the work done now by a former postdoc, and we were able to understand that without that vibronic mechanism and with the vibronic mechanism, the distribution of energy will be completely different in space. And it turns out that this compares with what it is observed in the experiment. Edward was able to show that you actually will have something really weird happening there, that you will have to represent this by a probability that has negative numbers, and that is a signature that you have something that you can never describe with classical mechanics. And this is by looking only at the vibrations. Just very quickly, and this one, I need to play this. We also focus on understanding this in a similar way that happens when metrons synchronize. So what you are observing there is on the left and on the right will represent the oscillators that you here have on the left and on the right. Here is at the quantum level, this is in the classical level. And when these oscillators are weakly coupled by this base, they start being no synchronized, anti-synchronized, and then eventually, you'll see the clicks, synchronized. Similarly, we're able to predict is happening at the molecular levels in this system. Of course, here we have a multitude of vibrational motions there, so it will be a synchronization of many of those degrees of freedom. But we were able to show that at least for this very simplified version of a light harvesting system, it is possible that many of these molecular motions will be synchronized in that way. This has important consequences because it means we have a collective behavior that we originally thought was only associated to the electronic degrees of freedom, but actually no. There are many vibrational motions in which this tends to work collectively to absorb and transfer energy. And this collective behavior can only be described with the rules of quantum mechanics. We were able to show as well that, okay, what if we take the inspiration from these experiments that are happening now? And is that we're able to take one of these molecules, connect it to a gold plate, have a little gold tip, and then we're forming a mini circuit. So this is really an experiment that they are doing with this. And I said, okay, I'm going to imagine the same experiment, but with a simpler system, and I want to know what happens if we have that vibronic mechanism. Does it improve the current that we can extract? This was the work of Richard Stones, and that's precisely what he predicted. If you have a complex enough vibronic mechanism, you increase the current, but if you reduce the complexity of that vibronic mechanism, that means if you reduce the number of vibrations that are contributing to that with a particular energy landscape, then you actually reduce the current. So again, it is showing us that that vibronic mechanism is something these systems are exploiting in a particular way. So I'm very excited because I think lots of experiments are now in the direction of proving how quantum is this vibronic mechanism and how we can exploit it. This is just an example of how it is being investigated directly in the biological system. And then what you would like to ask, okay, this is all very nice for photosynthesis, but what about all the other systems? Is the quantum biology only the quantum physics of photosynthesis, or is there anything else? So as I mentioned at the beginning, I love these systems because they have allowed me to understand many things that go beyond the biological molecules embedded in photosynthetic systems. And that idea of that collective behavior between electronic motions and vibrational motions can be translated to a more general idea of collective behavior between what you would call the relevant degrees of freedom and something that was thought to be irrelevant. So this collective behavior of the different degrees of freedom that are involved. Not too collective for too long, but collective enough for the time that the process is happening. This is the European robin. If I read correctly, in 2015, it was the bird of the UK. I'm not so sure, but I'm not a bird expert, but I read that. And this European robin migrates for many miles from Northern Europe towards Spain, south of France. It is not well known exactly how that migration occurs. What is the mechanism that guides them? Normally, they don't fly in flocks, so they are individual migrators. So what is what they are doing? What is the mechanism? One of the hypotheses is that they are detecting the weak magnetic field of the Earth. Not just the strength of the magnetic field, but they are detecting the direction. And by detecting the direction of the magnetic field, they are able to find where they are going. Now, one of the hypotheses is that this process happened because in the retina, there is one particular protein that supports a process that now involves electrons, but a different degree of freedom of the electrons, which we call the spin. So that intrinsic rotation, let's see it in that way just for the purpose of this talk. And these spins of the electrons have also interactions with the spins of the nuclei. What we are starting to understand is that all the interactions between these spins promote a collective behavior, which we thought it wasn't like that. We thought the nuclei and those spins in the nuclei will completely destroy any quantum behavior of the spins that are collective of the electrons. So this hasn't been the field of my expertise, but we wanted to understand whether that idea of that collective behavior was also happening here, whether that collective behavior was actually helping this mechanism to detect better, not just the magnetic field, but the direction. And that's precisely what TK, a former PhD student who finished last year, demonstrated. What this figure was showing is that TK considered many different possibilities of those spins, many different interactions, but the common theme to all of them was to have that collective behavior. And when that collective behavior was larger, we saw that the sensitivity with respect to the direction of the magnetic field was larger. So again, the idea, and this was an idea that was put forward before. What we did here was demonstrate that it was more general than we thought. So, to conclude, what can we do with all this knowledge? What can we do with all these theories that we're building? The first thing that I would like to do is to make sure that the experience we're trying to generate with our group in Glasgow confirm many of the things that we are predicting theoretically. You will know if that happens. But it's not just what we are trying to do, it's what we as a humanity are trying to do. The sun belongs to no one. And with the crisis of energy that we have so far, wouldn't it be so nice to have access to solar energy as much as possible? Now, there are many industries trying to improve solar cells and trying to create alternatives to exploit solar light. I do believe that the knowledge that we are gaining to understand the photosynthetic systems will eventually help us to create novel technologies, improving our capacity of exploiting the light that is coming from the sun and then meet our energy needs. But we also have an interaction here in biology, which means that we can modify these systems at the genetic level, and therefore the merging between the genetic manipulation that we can have of these systems our ability to understand them and manipulate them opens the possibility for having systems that could be used for therapeutics, for many different implementations, such as, for instance, putting chromophores in DNA materials, and many more that I don't have the time to tell you, but that I can tell you during your questions. Thank you very much.